Okay, so this one we are going to do, pen doesn't work, come on pen. We're going to do amino acids and proteins. Now remember, amino acids, when you put them together, you make a protein. So if you've got separate amino acids and you do a condensation reaction to join them together, you will get a protein. So a simple chain of amino acids would be a polypeptide chain. Once you've folded it, that's when we tend to call it a protein. So let's have a look at amino acid. You have a central carbon. Now that carbon can form 4 bonds, as we already know. Off that central carbon, as the name suggests, you have an amine group. So that is your amine. And on the other end, you have an acid. So that is your amino acid. Now also coming off that central carbon, you have hydrogen, always, and then you have something called an R group. Now there are 20 commonly occurring amino acids, which means there are 20 different R groups. It's the R group that makes an amino acid a particular amino acid. For example, glycine has an R group which is just a hydrogen, but you can get all kinds of things in those R groups. Okay, so that's an amino acid. All amino acids have that basic structure. You have a central carbon, you have an acid group, an amine group, a hydrogen, and an R group. So what we're going to do is we're going to do a condensation reaction and put 2 of these things together. Now remember, condensation reactions are always the same. You are condensing, you are squishing things together, you are removing water. So we are going to have our amine. You could just write that like that if you want to. We've got our central carbon. We've got our R group and we've got our hydrogen, and then again now what side you put the R groups on, it doesn't matter. Obviously chemically, if you kind of look at this, they'll be where they need to be to kind of fit in, I suppose. So we're going to do a condensation reaction. So we already know we are going to be removing water. So again, this is nicely lined up to easily be able to do that. So we know we're going to be removing 2 hydrogens. We have 2 hydrogens nicely lined up there, and we're going to be removing an oxygen. So when you've removed those, all you then do is bond them together. Now most of the time you'll have the double bond O on one side, the hydrogen on the other side. In your exam, it doesn't matter if you draw that, you will get the marks. Okay, now that is called a peptide bond. Okay, so that is a peptide bond. The test for a peptide bond, so therefore the test for a protein, but it doesn't work with amino acids. You can't use this for amino acids. You can use it once you've condensed some amino acids together. So the test for that, you add biuret reagent, and it will go lilac or purple. Okay, so that is the test for a protein. You add biuret reagent and it will turn from blue to lilac or purple. Now just a tip, I suppose, for any of these food tests, any observation you're making, don't just say stays the same. Tell the reader what it stays blue. If it has changed, it's changed from blue to lilac purple. Always put full observations into a table. It shows the reader you really do know what you're talking about. So if we wanted to break that hydrogen